All right, let's get started. So today we're going to be kind of going into the chromosomes. It's going to be this lecture and it's the one we come back and forth. We're going to be connecting on. But when we start thinking about it, like within our DNA, we can guys do the whole transcription. If you guys saw it, there's a lot of information that's kind of stored in the and we have to start it out in order for us to make it a new class and stuff. So we have 100,000 genes, which are crazy amount of exons in ways. So then, if you're interested in this, imagine that in one most like that equivalent, you have like 100,000 different cookies. And that is like, that would be so much overwhelming data that you probably find a recipe from those sets. And that's this really cool thing of our DNA, depending on what cell we're going through, they're going to have slightly different dynamics. Right? So instead of having all 100,000 cookies, maybe they close 99,000, leaving 1,000. And like within 1,000 books, that's a manageable library, we'll be going to in that section. So today's lecture, we're going to be looking at how we can actually organize these things to condense some data, put them kind of basically away from storage, while leaving other things open and other things we can actually when we come back from break, we'll go over the mechanism of basically these little yellow things when we fall in and figure out histones. That's 8 proteins that go this thing called the nucleosome. And that basically, for every 8 proteins, you have like 2.4, but it's more than like a little bit more than 2 laps DNA around this molecule are going to basically be trapped in it. And then we can use this to kind of further stack. Right? So you can imagine here, this is our DNA. It's 2 nanometers this way, like width-wise, and then length-wise. I'll give you the numbers again. And I can see how far apart they are from each other. You'll see like it's super short, but our stuff is so long, we can have like a meter of DNA. So then you'll see first level, we're asking around the histones, right? So that protein complex of DNA and the protein, that's what we kind of call the nucleosome. These nucleosomes right now, they're drawn pretty simply. But once we go into today's lecture, you're going to see they're a little bit more complex than just the spheres. These will kind of stack into themselves. This will kind of form little loops into themselves, and then these will kind of like curl and twist and turn into each other to eventually give us this, which is going to be our chromosome. Now to check out how impressive this is, here on top we're basically showing the human chromosome number 22. Right? So if we're looking at that, let's say this is one chromosome, this would be like, for example, like what I'm talking about, your mom's copy, your dad's copy, right? This would be the one that you can get from your mom, and the top one would be basically the copy that you get from your dad, and that's how you basically make your homologous chromosomes, right? In this case, we're zooming into just one of them. You have 23 from each. We're zooming into this little part, and then from there, we're looking just at 10%, right, of that little fragment. And within that 10%, we're going to find 40 genes, right? So if you think about this, that technically means that each chromosome might get us about 400, and then you have like 400 times the 23. You see, then this is getting bigger and bigger. Right? As we zoom in, we see basically this, the different reds, the dark reds and the light reds, they kind of represent the different genes. When we zoom in another tenfold, get a little even closer, now we're looking at just the central chromosome, and on average, or like we're probably going to find an average about 4 genes. We might vary a little bit, but this is something kind of what we'll find. And in that case, right now, we're zooming in another time, and as we're zooming in closer, then this just starts looking a little familiar. Right? So this is what we've been talking about the whole time, that these are regulatory DNA sequences. These are basically, they're really, really fancy versions of promoters. So that's going to be a code that's on the DNA that tells us things when, where, and how much of whatever is on that side to be made. Right? So let me draw some outside view of about it. So when we first started out, you guys only need one promoter, right? It was the basic promoter, so you guys need your top box. Which again, that's our DNA. It's in that code TATA8. That's the sequence that a molecule, transcription factor, knows to come grab onto this, and it decides to go back. In this case, that yellow region is actually a lot bigger, and there's additional sequences like the one my molecule likes to work with. Love that sequence. So TAAT, it would grab a bunch of these in a row, and it would basically link up with this, and that's how it would tell this signal to come on. So the job here is a promoter equals DNA binding site. And its function is when, where, and how much the transcription. But that's the big control that you have now. This thing is going to be passing down this information. Just how, remember, you guys go from the powder box, bring in the polymerase, and start making RNA. These things are super fancy. In each cell, if the molecule that binds to this is not present, nothing's going to happen. Only when this thing shows up, we get this thing to work. So these are, like the one I worked with was about like 3,000 base pairs, and then I went down to, we were lucky to find that only one base pair was the most critical one. I could switch it, and I would mess with this thing like crazy. But, right, so this will be the thing we're looking at. This is a lot bigger. Next, we're kind of going into it. You can see this little red would be your 5' untranslated region. Right. Remember, the moment you start making an RNA, it's about 10 bases after you see a powder box, your mRNA is going to start moving. We'll call this a pre. Pre-mRNA, right? So it's mature. It still has the introns, and you can just splice those guys out. But this little part here, so from the beginning of your mRNA, remember your ribosome reads into it, and it won't start translating until we hit that first AUG. And then it kind of keeps going, right? So then technically this is a 5' untranslated region, right? So we're UTR. All right, this will give us the rest of our gene. And here you see they're also giving us, so there's the familiar stuff. There'll be your red exons, there are your introns. We'll throw an intron here in the middle. And we'll do one more exon. And then here we'll have our stop, right? And after that, our RNA is still kind of a little bit longer. You can have to probably detail. And then we call this our 3' BC, right? So you can see all that information. These things could be way bigger. I've seen some of these in like the thousands of base pair. Some of these I've seen them as small as like 30 or 40 nucleotides. And then that's the smallest exon, like a really small exon you can bring in. Then you see all this information is all basically stored and will be considered like 1% of the chromosome. And this is just for one gene. And then there's some really fancy maps that they do now where they draw this whole chromosome, kind of showing this little thing of information. In bioinformatics, I can zoom in and actually look at the code. If I keep zooming in, I can actually see the base code with the ATG, the stock codon, and how they function. But the crazy thing is, right, from that little stored place, we're going to get it from, we can have it in two different places. We can have it or closed up, or we can have it opened up. And then that super upstream event is going to tell us will we be able to activate this path, right? If it's closed up, you're not going to bring in the polymerase, sorry, the transcription factors, you won't bring in the polymerase, you won't make RNA, you don't make RNA, you can't make protein, you can't make protein, you don't have it. Yeah? So it's going to be, it's a super tight control mechanism. So in this case, what we want you guys to know is like, when we talk about the genome, we're talking about in organisms, like all the DNA. So we're looking at all the chromosomes at the same time. Right? Within that, we can do this thing where we study the DNA proteins, and that complex, both of them together, is what we call the chromatin. So that'll be our histones inside the nucleosome when it's wrapped around with the DNA. And then I think we've talked about before, our chromatin varies in degrees of condensation, right? So we have our two states. What's the first one? What are they called?
Euchromatin and heterochromatin.
Mm-hmm. All right, and then what is the, let me pick on this side. What is a heterochromatin? What do you know about it? What do you remember?
It's tight and compact.
All right, yeah, hetero, it's going to be tightly packed. So we're looking closer to this side of the spectrum, right? And then for our euchromatin,
It's loosely packed.
loosely packed, right? So we're sitting here on the right side. Like this, we can get the histones to kind of fall off. So there'll be our two little states that we can kind of play around with. And normally the heterochromatin becomes super important for this. You can see here we're looking at different cells. In purple, you have stains of DNA. And what you're looking at is while the cell is still like living its life, doing its thing as a single cell 2N, you're going to do something like this. The DNA is going to be spread around that nucleus. Some parts are going to be open, some parts are going to be closed. But then this cell and this cell are going through different stages of mitosis, right? This is the time where these cells double up and they have to divide. And when you do that division, both of your daughters from the same mother, you divide them into two, they have to have all the DNA. So the genome that you find in this cell, you have to make 2 copies of it, and then you have to split them evenly between the 2 daughters, like this. So this is where we're going to see that there's really cool fancy tricks where we'll get a chromosome, we'll make an exact copy of it like that. Then our cells really know how to grab onto these and they'll pull them apart. So each daughter can get a copy of each single chromosome. All right, so that's a super important place for the chromosome. All right, so again, I'm trying to feed this story to you guys that will be connected all the way from the top. So I kind of like this drawing a little bit better than the ones we've been getting in our book. So for this one, you can see all the way at the top, there's our chromatin status, right? Or our chromatin. And again, we can have this thing be existing in 2 different states, right? Heterochromatin or euchromatin. So I'm going to be closed in the hetero, open in the eu. If it's open, then we can follow down this pathway. Technically, if it's the heterochromatin, I would put it there, kind of go out to the side and be like, you're basically stuck right there until something happens, right? So open chromatin, there's our DNA, and again, there's our storyline, right? Transcription, we make our pre-mRNA, we have our caps, our tails, we're gonna get rid of the introns through our RNA processing. Now that you have your mature RNA, send it out to the cytoplasm. And there, we're gonna focus on this branch, which is you have translation, right? Ribosome's gonna get us our protein, the protein's gonna get further modified, and you can start sending it out to different organs. So this little this whole decision thing of like, do this, do this, do this, do this, do that, we're doing it on different regions of this chromosome, right? And the cell is somehow being able to pick, like, I'm gonna use this recipe, this recipe, this recipe, I'm gonna skip everything in between. And when you take out a nucleus that's in like let's say midlife, it hasn't divided yet, it's still, just trying to maintain itself, you'll find something that looks something like this, which is super cool. And there, what you're seeing is on this side, this darker region, that represents our heterochromatin. Right? In that place, we have our DNA tightly wound. This thing is stored up. And then we have this thing, these little like swirly lines that we have coming out like this. Those are basically the thinner strands on this side. So meaning that there's your heterochromatin, and we're starting to look towards our euchromatin, where we'll start making move proteins off this thing to kind of open them up more. more so we can start using it. And they make these like super cool little structures. So how's the chromatin working, right? So we're gonna have to make the decision, euchromatin or heterochromatin. How does the cell do that? So one thing we have to look at is gonna be our histones, right? And what you're gonna see is that histones, they can reversibly bind to DNA. And by that, I mean, you can have DNA coiled around them. You can also have DNA let go of them. They also have these little arms that, depending on their charge, they can affect how well they stick to things. The DNA has normally a negative charge. So if the arms have positive charges, they're really good at sticking to the DNA. When the arms have negative charges, they'll kind of repel off each other. So by us controlling the charge in the arms, we can control whether it's going to be wrapped tightly or loosely. And this is where there's going to be a lot of other proteins that are going to come into this picture. So there's going to be different ways that we can actually try to beat this competition. So in some cases, there are some proteins, let's say like this little brown one right there, they're called pioneering factors. In those cases, even if our DNA is coiled tightly around the histone, a pioneering factor doesn't care. If it likes the sequence, it'll go in there and it'll grab onto it. So even if you're in like full heterochromatin, this thing is tightly wound, this molecule can squeeze in and grab onto its site, and it'll bring in more partners. So here I'm kind of showing that one as a little purple one. And then these partners, what they'll do is they'll help modify the histone. Right? So you can imagine this thing's closed up, pioneering factor shows up, we bring in the other proteins to help us out, and we start modifying the histones, and basically it'll push them away. So a place that used to be a heterochromatin, we can go through that pathway, basically open it up. Right? On the other side of that, we're going to do, there's other protein complexes that do quite the opposite, right? What they'll do is they can bind to free DNA. They'll bring in, we'll play it off with this one. So that blue one, it can bind free DNA. It has the ability to interact with other molecules that have the opposite effect on the histone. So if the modification made the histone less likely to wrap around DNA, this thing will go, and any histone that gets near it, this stuff will basically swim by, it'll modify it so it wants to grab onto DNA. So wherever the blue thing is, you're going to end up having an enrichment in that area of histones that want to get coiled up. And that means that what used to be a euchromatin, we can then walk it back to make it a heterochromatin. That make sense? Yeah? All right. So there's a lot of proteins, a lot of fun stuff going on. And we got some pretty cool ways to kind of look at it. All right, but now let's zoom in, and we're actually going to look at the nucleosome inside of it, how it's built. So for this one, the main body of it is built from 8 different proteins, right? So what you end up having is you have two sets of 4. So this would be 1, 2, 3, 4, and on this side there's another 1, 2, 3, and there's 4 on the back. So these will be the 4 proteins that you have. The naming convention, I hate it because H2A, H2B, H3, and H4. Right? These will be our 4 different proteins. These kind of like bigger chunks that they're showing you here, those basically what's going to give us the main body of it. So this thing right here, right? That little triangle part, it'll be the core around where we need stuff to wrap around. And I hate this naming convention, but N-terminal tail. So N-terminus means this is the front of the protein, but tail, they kind of refer to this little thing like this. So this little, you have little arms that are kind of sticking out from the histone itself. It'll have its N-terminus sticking out. And this is where we can do a lot of modifications to make things happen. Right? So if you look at these, when you're looking at this picture in B, you're seeing that these basically, the tail from one histone, look at that, it leads to the interaction with other histones, right? So it's like, hey, if you have the right hydrophobicity, you have the right charge, it'll be possible for one histone to very happily grab onto a neighboring histone, put it close. If we start doing modifications on this arm, we can do the opposite. This interaction is no longer going to be favorable. It's going to let go, and the histones basically open up. So by controlling the arms, the tails themselves, we can control whether it's going to be a tight interaction or a loose interaction. And technically, these are Non-covalent interactions, so again, we have a little bit of hydrogen bonding, we probably have a little bit of van der Waals. So remember, super weak interactions, but just how we showed the magic in water where hydrogen interactions add up to have this huge powerful thing, that's what's going on here, right? Super weak interactions, but a bunch of them happening would give us something very strong. All right, so we're zooming in even a little bit more. So here you're looking at the different tails, sorry, the N-terminal tails. And for this one, these are basically the modifications you're bringing in, right? You have your methylation, phosphorylation, and acetylation. When you bring an acetyl group, right, like this one, when you, the little A's, it was showing you that's your first amino acid. I can see right. Let me double check this. But at position 1 of this tail, you can see that S is able to accept the phosphate group, right? When you bring in a phosphate, you guys remember, from our DNA, it's going to look like this. So you're going to be bringing negative charges, right? So we said the serine right now, I'm opening up the periodic table shape, sorry, the amino acid table so you guys can see what it looks like. But then you're bringing in this thing with a huge negative charge. And again, our DNA is negative. It wants to interact with things that are positive. You put negative and negative, they'll push each other away. So at this amino acid site, we could potentially either have it phosphorylated or not phosphorylated. And there's proteins that do both things. Some of them are called kinases. They'll give us a phosphate group. And then phosphatases will take a phosphate group. The other one that's kind of cool is going to be this one, this acetylation. So there's your acetyl group, right? Which is basically you're going to be bringing in like carbon with a double bonded oxygen. If you look at it at the K site, there's a K site there. At that site, you have basically K means it's a lysine. There's your N-terminus, central carbon. There's your carboxyl, right? That keeps growing the chain of this thing. And there's going to be our R group, right? Like our side chain. At the end of this, you see we have a positive charge. That means we have something that's hydrophilic. And then this is basic or acidic. All right. If you put something positive into again, to interact with DNA, our DNA is going to be happy, right? It's going to want to stick to it. The moment you put in a acetyl group, you're taking away that positive charge, and you're putting in an oxygen, which is super electrolytic, right? So you literally just change the dynamics of that part of the arm. So just like this, you can switch this thing back and forth. This modification is reversible, acetylation. So you have acetyl-CoA. So acetylation coenzyme is the one that helps us jump from here to add the group on. And then we have the acetylation, which is, there's a few other proteins that kind of basically walk us back, and it will take this off and leave us back to this. So there's two proteins that control the fate, but our tail is going to be able to jump between the two states. Positive charge or like negative charge by that oxygen group. The methyl group, this one's going to be it's a little bit more complicated for that one because what you're doing is you're basically adding like carbon groups. Like methylation is basically just adding an extra carbon, so you have a carbon chain here. Sometimes it messes with this a little bit, sometimes it doesn't. So for that, what we're going to do is we're going to have, in reality, if you spoke to somebody who works in this area, they would tell you that the histone code is very complex. The modifications themselves, they're very specific. Like there isn't one general rule where we're like, hey, if the thing is phosphorylated, for sure, that DNA won't grab onto it. This methylation, there's some mutations where like, if it happens in certain states, if the methylation happens at certain arms or at certain spots, sometimes it has no effect. In some cases, it makes it better binding. In some cases, it makes it worse. Right? So it's like very specific on where the methylation happens, what's going to be the outcome. Yeah. So there used to be a lot of papers first in the field because we used to fight this idea off and they'd be like, no, acetylation is always a negative thing. And then somebody would be like, but if we look at a paper, H2A at amino acid number 9, if you acetylate that, it actually does the opposite, right? And that's what they were fighting for. But it is that complex. For the sake of the midterms, though, I want you guys to stick with this, right? So we're gonna be like, this is our simplified version. If I talk to anybody who was working on histones, they'd be like, no, dude, it's more complex, right? But this is what we're gonna use. Again, our methylation, we're adding just a carbon group. This one's gonna happen at a lysine. That'll be our Ks, right? I want you to think that the histone is not gonna change at all. So we're not doing that jump between heterochromatin to euchromatin. It stays the same. Next one, acetylation, right? So in this case, we're bringing in a lysine. Same thing's gonna happen to that same lysine. But now, you're actually gonna make this thing more negative. And the big thing is you're missing that positive charge now, right? So here I want you to be thinking, when we have an acetylation on a lysine, we're pushing towards not grabbing the DNA that well. We're not grabbing the DNA that well. We're walking towards the euchromatin, right? So this will get us towards the euchromatin. Phosphorylation, this one happens on a serine. And in this case, what we're doing is we're bringing in a stronger negative charge because we have all these little groups basically hanging around. And in that case, right, same thing. You have an arm. If it had even a little bit of positive charge or no charge at all, the moment you make it more negative, our DNA is not going to want to hang around it. So it's going to help loosen it up. So this is going to be another one that's going to walk us from our heterochromatic. And when we start modifying these, we're going to start walking towards the euchromatic. So let's see how I can do this here. So let's say we were at euchromatic. Right? This is kind of what I drew here in the class, right? If we're trying to walk our way to making this a heterochromatic, where this thing's basically folded up onto itself. So that's my rough drawing, right? The things that are going to be helping us out to get us down this pathway is bringing in those phosphorylation marks, right? Bringing in those acetyl groups, bringing in modifications. We're modifying the arms themselves. By modifying the arms, we can end up producing the change in how the DNA is going to be coded. Yeah? So here we're kind of looking at a super zoomed in version. So these are with that scanning electron microscopy. So we can actually look at these things. With the regular microscope, we can't get that far in. But here you're looking at, there's our euchromatid. There, this little string would technically mean that's our strand of DNA. And then each one of these little spheres are a little bit darker. Those are basically our histones, right? So the 8 proteins are right there, and our DNA is wrapped around them like this. But look, in this case, our lysine is being acetylated, right? So we're bringing in those carbon and that oxygen group. So you're bringing in that negative charge. Now the arms that before were happy interacting, grabbing onto the other histones, they're not going to want to do that interaction anymore. They'll separate from each other. But it also messes with the interaction back here. And actually, you can even modify them enough where it kicks them off from this thing. But yeah, so euchromatid, there's our marks. If we remove them, it'll walk us back up here. We regain our positive charge. Now we can actually be able to stick to itself. Questions? No. All right. So if we look at the acetylation itself, now we can actually look at the kind of how the chemicals are set up there. This would be the lysine. This is the one that you guys basically have seen a few times when we did all the amino acid work, right? So just to orient yourself, there's the amino group, the N. There's our central carbon, and this will be our carboxyl group, right? And then the O would basically be on that side, linking it to the rest of the chain. So for this one, there's all these carbons, but the important thing is going to be down here. That's your NH3 ring. That's what's going to give us that positive charge. So in this case, here you can see that lysine that we talked about before, there's 2 modifications that can happen to it, right? One of them is we can go this way, and we're doing that monomethylation. So you're bringing in that little carbon right there. When you do this modification of giving it a carbon, this still has a positive charge. Like that is actually kept still, right? So this is technically, it doesn't have an effect to the binding because this thing is still gonna be positive. The opposite mark is gonna be this one on this side where there's our positive charge, there's your 2 carbons with a double bonded oxygen, which is our acetyl group. When you modify this, you've taken away that positive charge and you're bringing this thing into the picture. Now, the reason it's important to put these two next to each other is that they kind of fight each other off. So only one can exist at a time. So either they're going to be in this state or they're going to be in this state. So you can imagine a DNA that, like our cells, let's say, haven't decided yet whether they're going to open or close it, is more likely going to sit right here as a lysine. If the cell decides, hey, I need to work with this DNA, you add an acetyl group to that histone, you're going to modify that histone, you're going to open it up, it's going to help that area stay open. Then if you have a competitor who's saying like, no, we want to actually turn this place off, like make sure seal this information off, you would go this way, right? You add the methylation, that's going to be the modification that's going to give you that more like, hey, make sure this thing's sorry, that thing's tight bound, this one would open up, right? Make sense? All right. So they're a pretty cool mechanism of fighting each other off. The other one we talked about is going to be this one, which is our serine phosphorylation. So again, amino group, central carbon, there's a carboxyl. When we go this way, that's the important thing. Serine and threonine, they look almost the same. I think threonine is just one extra carbon, and this OH is like a little bit down here. But that free OH at the end is the place where we can let us do this and bring in our phosphate groups. So when we bring in the phosphate groups, look, oxygen, 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 you're going to bring this area that's super negatively charged. And this is where adding this thing, it's going to want to kick the DNA as far away from this thing because it's not going to want to interact with it. So super powerful stuff. Then there's also a little bit of information that we kind of find that it's also coming from the DNA itself. In this case, we're kind of showing you that you're imagining we're kind of looking at a side view. This is the top of your histone, and then this is the DNA as it's doing the loop around, right? So the DNA, when it makes the double helix, it looks like a coiling ladder, right? And when it's doing that, they call the space, you have like these little tight junctions, which these would be called the minor groove. And then here you see there's a big space between this to this region. That's basically your major groove. Right? So you have that weird coil that kind of does this. And what they find is that the stuff that likes to bind, what's it called, with the histone itself is going to be TA-rich regions. And then your GC stuff is basically going to be kind of hanging out. There, I'm not sure, like in my head, I'm thinking that it's going to be, remember, our TA regions are the ones that they only have 2 hydrogen bonds. So that means that the purines and pyrimides technically have more open functional groups to basically grab onto. The GC, we have a 3-to-3 interaction, so you use them a lot more than the other one. I'm not sure that's exactly why it does it, but to me, that's a little thing that I kind of think that's what's giving us this effect, right? So, yeah, so kind of to bring it all together, right? Remember, we can jump on this thing, open DNA, we can get this intermediate form, we can condense it, further condense it, and eventually we get it all the way to the chromosome. Yeah, so this part I went through, I mean, case by case, right? I was telling you how do we change the acetylation, how it's going to affect DNA protein interaction. I told you about phosphorylation, how it's going to hurt us, methylation. On this next one is going to be all these changes aren't just happening randomly and they're not happening in general, right? So if you imagine the whole class to be like a huge chromosome region, each one of you represents a gene. It's not like all of a sudden we're bringing in phosphorylation and everyone's getting phosphorylated. The cell actually goes and targets and it'll be like, you're getting phosphorylated, you're getting phosphorylated, you're getting phosphorylated. We're going to open up those genes. Will be methylation up maybe on this corner or different spots just randomly, and I can turn those genes off. Right? And then I can also do the acetylation. The cell will pick the different genes and open those up. And that's done by basically, there's going to be these proteins that are going to be able to modify our histones. They help with recruitment. They help with modifications. They also can even help kick them off. Like they'll throw them away from the DNA to open up a region. I think I went pretty tight on that. Yeah. So again, the two different states, right? Compact versus open. I said that more than a few times. So here we're kind of looking at, again, putting it into the big picture of how we're going from this state up here to jump into turning the gene on or not having it on. And there would be these different proteins. So there we're looking at heterochromatid. Our DNA is super tightly packed. Your daughter box, right? That critical thing that we need to give the polymerase so it can actually start making this, that's going to be tucked away inside of the histone. The polymerase itself and the transcription factors it uses are too big to make it into that spot. So when it's in this state, technically the gene is off. We can't get into this gene off, no expression. Then you have these additional transcriptional regulators, but my favorite one is that one, the pioneering factor, because it'll come in, it doesn't care that this thing's closed up, and it can grab onto the DNA. It'll grab onto these little fragments, and then that one will walk us kind of down this pathway up here, right? It will grab onto the site, recruit these chromatin remodeling complexes, which are the proteins that are going to be affecting the acetylation, phosphorylation, and methylation. And as it's doing that, is basically that little green one is then modifying the different histones within that area. So you can kind of imagine if the pioneering factor landed wherever it lands on the sequence, it's going to bring in the molecules to help it. It'll open up that way, and it'll open up that way. So it'll be like open up. It's going to basically slide down till eventually this super compact form, it'll open up in that region. But just like there's, so there'll be signals that, let's say one gene, say open up this way. You can also have genes in other regions which are sending the opposite message. And it'll be like, close this thing down, right? And then it's kind of like just a little bit of a fight. Like maybe the neighboring gene says off, it'll be off for a few histones. This one's saying on, and they'll kind of meet in the middle and they'll stop there, right? So they make it these little borders of open and closed chromatin. So yeah, so once we have the nucleosomes basically sliding away, some of them are getting kicked off. The next thing you're looking at is this part will be getting us into this. Now that it's open DNA, you can bring in our promoter, our enhancers, those are promoters. That'll help us recruit our transcription factors, our polymerase, and start kind of helping us with gene expression on that side. And then here's the opposite route, right? Which is whether you're like, let's say you're in this intermediate phase, you bring in the histone-modifying enzymes that are doing the opposite to that one. And then the goal here is we're going to add more marks and going to close this thing down. This is really nice. Histone traces, stabilized compact forms. Oh, my bad, I said that one wrong. So you're modifying it and you're bringing in trap components, transcription factor. Yeah, it's my bad. So this is the modifications. This one's literally just the physical kicking them off. Like you can actually kick the histones right off. My bad. Change it up. So here put, like I'm almost thinking about it. Like, let me see what have you done. But it's like you kind of just grabbing the DNA and this thing just pushes across like that, and it actually makes the histones shift as it's kind of shoving the nucleosome forward. On this case, this is going to be the modification. So like the phosphorylation, and that's going to be the thing that's going to help loosen up the interaction. Sorry about that. So, so you can imagine there's going to be different proteins that help. So just like I was saying, you can grab that string and kind of like guide across it and push the histones off. There's also going to be stuff like this NP1, which is going to be a loading protein, and it's going to help bring these guys in. So there's your H2A and H2B, and they'll kind of be stacked on top of each other, and then we'll start putting this thing onto the DNA itself. That's going to help us go from an open DNA to start closing it up, putting it into the histones. Here's your little fun one, your sliding clamp. That's the one that grabs onto the DNA and glides across it. As it's doing that, it's the one that's giving us the force to kind of like a snowplow and just basically taking things off, right? And here they're kind of showing you in the middle of DNA replication. So this is a little bit of a replication fork. We haven't gone over this yet, but the DNA can make really good copies of itself. They're efficient and they're very, what are they called? They're robust when in the sense of mutations, like mutation rates are super low. So here you're imagining this red part is the side of the DNA that's coming from the initial mother cell. Right? It only has one copy of each chromosome, but it's trying to divide soon, so it has to make copies of it. So what it does is, in here there's replication fork, you have to take off all these histones, open up that area, and when you do that, you're actually separating the two DNA strands, and you pull them apart. One DNA strand will go this way, one DNA strand will go that way. When they're opening up, you have DNA-dependent DNA polymerases. So just like we had our DNA-dependent RNA polymerase number 2, POL2, right, which uses DNA to make RNA. Here we use an enzyme that uses DNA to make more DNA. We'll have one of them copying one strand, the other one will be copying the other strand. So technically you get one cable from the two cables of DNA. This one gets one cable and that one gets one old cable. And then in this area here, you're basically creating a new cable to kind of go with it. Right? And you're using the hydrogen bonds to perfectly match in your cable. After that, the real cool thing is you have this whole thing open. You're gonna start putting it into the histones. And the cool thing is you can technically pass down information, right? So if, let's say, the parental mother cell had a region of DNA and it was like, I want it to look like this, right? This thing has the ability to open this up, make it single-stranded, make itself 2 copies, but when it's putting the histones back, it'll bring in that type of modifications and put them in here already. So before there was a theory that they were thinking, they weren't sure if it was like, when the DNA was copied, did one, like were they getting like one histone for you, one histone for you, one histone for you, one histone for you, so the 2 different daughter cables, in order to pass the information. Some people probably completely reset, but for this one, there is this thing where it's like, if in the mother there was this case, they'll basically keep the modifications in that general area and they'll come back and further fill this in by adding that, and then we'll sort it there, but it will close up, keep the euchromatin, and the heterochromatin, it'll keep it a heterochromatin. If the daughters want to differentiate, then they'll start making little changes. All right, so how you guys feeling about today's lecture? Pretty good? Yeah? All right, so I'll see you guys on Wednesday for in-class activity. And then I will break.